Dear all, welcome to this video. Please hit the subscribe button so that you automatically get updates for upcoming videos and lectures. Thank you. Enjoy watching.
Cell-mediated and humoral immunity. Now these both immunity are caused or they are responsible because of the B lymphocytes. Sorry, because of the lymphocytes itself. And where these lymphocytes are synthesized? They are synthesized in lymph nodes, spleen. They are actually present in lymph nodes and spleen, but they are synthesized in bone marrow and thymus. So these are your lymphoid organs. So now there are two types of organs, lymphoid organs, that is primary and secondary. Primary are the organs where actually synthesis or manufacturing takes place, and secondary is where it stays or if the storage may happen. Okay, so primary organs are thymus and bone marrow, and secondary organs are lymph nodes, spleen, adenoids, tonsils, appendix, etc. So now this is the difference between lympho T and B lymphocytes. So lymphocytes are usually classified into two classes, T and B. So depending upon the site of production, they have been classified. All those lymphocytes which are produced, actually both the lymphocytes are produced in bone marrow itself. But after production, the maturation of these lymphocytes takes place via thymus. So those cells are known as T lymphocytes or T cells, and those which go via bursal equivalent tissue, those are known as B cells. Okay, so here you can see bone marrow stem cells. Okay, and those which are going through thymus maturation, those are T lymphocytes, and the other going through bursa, those are B lymphocytes. So T lymphocytes are responsible for the cell-mediated immunity. B lymphocytes are responsible for the humoral immunity. What are the secretory products? T cells act by releasing the lymphokines, whereas B cells act by releasing antibodies. Then in T cells, there are subpopulations or subtypes, which includes helper, suppressed, delayed hypersensitivity, and killer cells. Natural killer cells, which are discussed, and non-killer, non-specific. So those come under T cells. And in case of B cells, there are only two types: plasma cells and memory cells. What is the effect? T cell works against virus, fungi, protozoa, and cancer cells, whereas B cells works against bacteria, virus, which are present only in body cells. And what is the type of response? T cell usually leads to engulfment, that is phagocytosis, or cytotoxic effect, that is cell death, whereas B cell leads to agglutination, precipitation, neutralization, and complement fixation. Okay. So now here you will see the actual difference between cell-mediated immunity and humoral immunity. So as we all discussed, bone marrow, which is a site of synthesis of lymphocytes. So the cells which goes through thymus, those are T lymphocytes, which goes through bursa, those are B lymphocytes. Now when this T lymphocyte is exposed to some antigen, what will happen? This T lymphocyte will first get activated. On activation, it will produce clones, that is, it will increase the number of T lymphocytes. So clones of T lymphocytes will be produced, and these together will release lymphokines, which are responsible for cytotoxic effect, that is the cell death. So this is known as the cell-mediated immunity. Okay. On the other hand, when B lymphocytes are exposed to antigen, what will happen is again B lymphocytes will get activated. Now after activation, the B lymphocytes will get divided into two, that is plasma cells and clones of B lymphocytes. Okay, and this process of differentiation, it usually occurs in the presence of activated T lymphocytes. So you can see there is a word called as cooperation. So even these are required for the differentiation process. So the plasma cells which are produced, they will form clones and then they will produce the antibodies. So that is responsible for the humoral immunity. And these clones of B lymphocytes which were produced, which these are known as memory B cells, and they will remain stored in the memory of host cell. And on second exposure, these memory cells will produce antibodies and kill the pathogens. Now next is the humoral immunity. Now this humoral immunity, we will just briefly introduce. We'll just briefly discuss what it happens is. The important points I'll discuss. So it is resistance against only bacterial pathogens and viruses that attack respiratory and intestinal tract. B cell is destined to produce only one antibody, though it can produce more than one class of antibodies. Okay, if it produce means one B cell will produce only one antibody. Okay, then B cell must receive a signal from the interacting. As you all know, T cell or T macrophages cooperation is required for differentiation and proliferation. And usually, what will happen whenever the plasma cell produces antibodies, after the production of antibody, it will die. Okay, and the mature plasma cell can produce a unique class and subclass of IgG. So this is the normal response. So whenever you are given a vaccine, say for example, antigen is given here, you can see antigenic stimulus. So antigen is given and you are calculating the amount of antibodies produced with the respect of time. So initial stage मध्ये, the number of antibodies will remain constant after a particular period of time, say 5 to 10 days. This will go on increasing. Then there again stationary phase will occur and finally decline phase. So this A is the latent phase where obviously time takes for production of antibodies. B is the rise in antibody phase. So it usually will happen, it will cause increase in the number of antibodies. In C phase, obviously the number of antibodies will remain constant and finally it will decline. So this is the primary immune response. Primary immune response here, it means that once you are vaccinated for the first time, whatever response you get, that is primary. So this secondary response is also called as memory or booster or anamnestic response. So you must have heard for some cases, usually in children, first dose is given, then the second dose, which is also known as booster dose, is given. So what is the reason behind it? We will see it here. So the first dose is known as the priming dose, whereas the second dose is known as the booster dose. And if you are giving a killed vaccine, now vaccines are of different types. So among them, there is live vaccine and killed vaccine. In case of killed vaccine, what you do is you give the bacteria or the particular virus is killed. It is not active, it is not pathogenic, it is completely dead, and that is inserted into your body. Why? Because the surface contains antigens and that is enough. So that is known as the killed vaccine. So if you are giving a particular person killed vaccine, then usually booster dose is required. And negative phase occurs. So why this negative phases occurs is because that particular bacteria will remain in your body say for a certain period of time. After that particular period of time, what will happen is that will be removed from the body. So continuous stimulus will not be provided by the body to produce antibodies, and these antibodies will not remain in your body for a long period of time. So it may lead to negative phase. But if you are giving a live vaccine, there it does not happen, and the reason behind this is because you are giving a live bacteria which has been attenuated, whose virulent strength has been reduced, and then you are inserting it into the body. So what will happen is it will keep on multiplying after a certain period of time, and the antibody production will also remain continuous. So in that case, booster dose is usually not required. In case of cell-mediated immunity, it involves T lymphocytes. It is useful in protecting against fungi, viruses, intercellular bacterial pathogens, intercellular parasites, rejection of foreign tissue grafts, delayed hypersensitivity, and resistance to some cancer. So response starts with T cell activation. We have already seen the cycle, so I don't think it is required to repeat here. Okay. Now these T lymphocytes, as you also see in the subpopulation, so helper T cells, suppressor T cells, cytotoxic T cell, delayed hypersensitivity T cells, killer cells, and natural killer cells. And these are their functions. Helper T cells are necessary for activation, B cell activation by T-dependent antigens. Suppressor T cells regulates the immune response and helps in maintaining immune tolerance. Cytotoxic T cells, it destroys the target cells. Delayed hypersensitivity T cell, it provides protection against infectious agents. Killer cells attacks the antibody-coated target cells, and natural killer cells attacks and destroys the target cells. So here we will stop. The specific immunity has been covered. In next lecture, we'll be covering what are antigens and antibodies and their structure in detail. Thank you.